Well, thanks everybody. I really appreciate you taking the time to come and listen to me talk about my research for a bit. My thesis is on plant-based recovery of rare earth elements, assessing the feasibility of phytomining in Idaho. Before I go any further, Dr. Hung, can you hear me okay? Yeah, okay, perfect. Just wanted to make sure since you're on Zoom. All right. Little bit about me before I actually get into the presentation. I earned my Bachelor of Science in Biology, concentration in Ecology and Conservation Biology in 2023. And I'm hoping to earn my MS in Environmental Science in 2025. I'm married and I have 7 pets, so it's never really a dull moment in my house. All right, moving on to the thesis outline. I'm going to start with an introduction into what phytomining is and why it's important. Next, chapter 1 is a literature review covering phytomining as a plant-based mineral extraction solution. Chapter 2 covers extraction of rare earth elements from Idaho source soil through phytomining and conducts a case study in central Idaho. Chapter 3 looks at phytomining of rare earth elements using native hyperaccumulator plants and surface soils from Idaho. And then we're going to finish with a conclusion and suggestions for future research. All right, so demand is growing for products and technologies that are made with the rare earths. These are the elements shown on the periodic table here in green. So those are the lanthanides down here, as well as scandium and yttrium up here. And these elements have really unique properties that make them essential in the production of things like defense and homeland security technology, cell phones, batteries, and then a lot of green technology, so solar panels, wind turbines, and hybrid and electric vehicles. Unfortunately, traditional mining practices like open pit mining, underground mining, and on-site leaching really threaten the environment, degrading its natural resources, biodiversity, and threatening human health and well-being through pollution and land degradation. So this middle picture here shows an example of the sheer amount of land degradation that traditional mining can cause. And then this is an example of some pretty severe water pollution. So why is this important? Well, there are select soils in Idaho that contain really high levels of the rare earth elements. Specifically, this Idaho rare earth thorium belt shown here that is on the border of Montana in central Idaho. I specifically want to draw your attention to the Diamond Creek area here. That contains about 1% rare earths within its soil. And that creates the unique opportunity for phytomining, where these special plant species called hyperaccumulators are grown, and they are able to extract really high levels of the rare earths from the soil. That biomass can then be harvested and processed to extract those minerals for industrial use. Benefits of doing this at a large scale commercially in Idaho, including that it would supplement rare earth metal production. It would provide economic growth for the state through things like job creation. It's very cost effective as compared to traditional mining practices, and it can actually be used to supplement traditional mining as well. And it improves soil quality for future use. So the soil can then be used to grow crops, for forestation, or to seed metal intolerant native species that can increase local biodiversity and help avoid erosion in the area. So the overarching goal of this thesis was to assess the feasibility of phytomining as a mixed rare earth extraction technique on Idaho-sourced rare earth rich soil sourced from that Diamond Creek area that I just mentioned. This was done through the exploration of various species, varieties, and treatments to maximize biomass and rare earth yields, as well as to evaluate the environmental and economic impacts at a local scale. So the objective of chapter 1 was to identify the literature gaps and challenges related to plant-based mineral extraction of valuable minerals. This was done through a narrative and systematic literature review, and currently the manuscript is under revision at the journal Renewable and Sustainable Energy Reviews. So this figure here shows the primary steps that are involved within phytomining. So the collection and characterization of the mineral-rich soil, the cultivation of the hyperaccumulator plants and the harvesting of their biomass, and then the heating of the biomass to produce biologically sourced ore or bio-ore, and then the digestion of those ores to actually extract the minerals and then put them toward industry. The narrative review covered a pretty extensive list of topics, starting with the phytomining of critical and other minerals. This table here shows the minerals that were covered within this section. The middle column shows the hyperaccumulation threshold of each mineral, and that is the amount of a mineral in micrograms that a plant must contain per gram of its dried tissue to be considered a hyperaccumulator of that element and therefore appropriate for phytomining. And then the right column shows some example species that are good hyperaccumulators of each of these elements. Some of them might be familiar to you. Next, the narrative review covered the sustainability aspects of the process, namely the environmental and economic impacts. And most uniquely for this paper, it covered the role of machine learning and artificial intelligence in improving process efficiency, addressing challenges, and guiding future commercial applications through things like aiding in species selection, yield prediction, and sustainability modeling. So the systematic review looked at the progression of recent phytomining research, finding that since the year 2000, research interest in plant-based mineral extraction has significantly increased, especially since 2018. So this leftmost figure here shows the increase in number of citations related to phytomining since 2000. And this figure on the right shows the increase in the number of publications that were focused on phytomining or related fields. The review also found that sustainability-related studies are far more common than economic or machine learning AI-focused articles, as you can see in this middle figure here. So this increase in interest to plant-based mineral extraction could be due to a variety of reasons. One of them being the kind of worldwide recognition of just how much traditional mining degrades the land and contributes to climate change. Another is the increasing adoption of green technology and resource retrieval methods. So methods of process improvement that were identified include the use of hyperaccumulation inducing amendments, including citric acid, ligands, and fertilizer. Another is the use of selective plant breeding and genetic engineering techniques to yield a superior hyperaccumulating line. And then the integration of machine learning and AI into various steps in the process, as shown on this figure here, including operation establishment, cultivation, processing and extraction, as well as the overall sustainability modeling. Challenges that were identified include plant growth time. So plants obviously can take a very long time to grow, and a lot of time that growth is very seasonal, depending on your geographic location. Project lifetime is also a considerable challenge, as over time, metalliferous soil will deplete and it just won't be economically viable to phytomine at that location anymore. Species selection is another issue. You need to pick a species that is going to produce a lot of biomass, going to be an excellent hyperaccumulator, going to grow very well within your chosen climate conditions. But you also need something that isn't going to damage the surrounding environment through things like becoming invasive. And there are also very few known hyperaccumulators. So out of all the ones we know, there's about 700 species that we know of, and the vast majority of those are nickel hyperaccumulators only, about 500 of them. And that leaves a lot of other elements, such as the rare earths, very underrepresented within the current hyperaccumulator database. And then also post-harvest processing is very underdeveloped currently. So it's going to require a lot more research before it can be scaled up to the commercial level. Benefits that were identified include the fact that phytomining is very environmentally friendly and it remediates the land. So again, the soil can be used for other things. Plants capture atmospheric carbon and store it in their biomass, so the process is inherently carbon negative. Excess biomass can be used to produce heat, energy, and biofuels. The process is very cost effective as compared to traditional mining. And there are opportunities for intercropping where non-competitive hyperaccumulator species are planted alongside more traditional crops. And so the land can be used to produce both your metal crops and then your traditional crops. So to conclude for this section, phytomining was found to be an environmentally friendly and sustainable method of resource extraction and can contribute to worldwide mineral supply. However, the upscaling to a commercial level is going to require a lot more research and development before it becomes feasible. The continued advancement of phytomining as a field is going to depend on its collaboration amongst other fields, including plant science, materials science, and data science. Recommendations for future research include identifying more hyperaccumulating species, further exploration into the genetics of hyperaccumulation and breeding efforts, and more study into the integration of machine learning and artificial intelligence into the process. Moving right on to chapter 2. The objective of this chapter was to evaluate the feasibility of Idaho-based rare earth phytomining by exploring the effectiveness of various species treatment combinations and assessing the environmental and economic impacts of a local operation. This was done through a greenhouse phytomining experiment, life cycle assessment, and techno-economic analysis. And this manuscript was published earlier this year in the journal Sustainability. For the phytomining experiment portion, plants were grown for 6 weeks from germination in the Idaho-sourced rare earth-rich soil from Diamond Creek. Pots were arranged in a complete random block design with 4 unique species shown in this figure here: Phalaris arundinacea, reed canary grass; Solanum nigrum, common name black nightshade; Phytolacca americana, common name pokeweed; and Brassica gensea, common name brown mustard. There were 3 unique soil treatments used: non-treated soil or soil treated with either biochar or fertilizer; 2 unique water treatments, non-treated water and 1% citric acid solution; and then 3 replications of each unique species soil water combination to total 72 pots overall. And as you can see, these plants were cultivated, then they were harvested once mature. The biomass was then washed to remove any excess soil that could possibly interfere with characterization. Then it was oven dried to remove as much moisture as possible. Biomass was then pyrolyzed or heated in the absence of oxygen at 400 degrees Celsius for 30 minutes. Resulting bio-ore was then acid digested using the EPA 3050B method. And then the resulting digestate was characterized via inductively coupled plasma mass spectroscopy. So this top figure here shows the inputs and outputs of cultivating the plant and pyrolyzing the biomass. This bottom left figure here shows the bio-ore that results from the pyrolysis step. And then the bottom right figure shows the digestate that results from the acid digestion step, and that's actually what is put through the ICP-MS unit for characterization. So once data was collected, the bioaccumulation factor of each plant was calculated. And that is the ratio of mixed rare earths in the dry plant tissue to those in the soil, with a higher bioaccumulation factor indicating a better ability to extract and store the minerals and better for phytomining. Next, 2 life cycle assessments were conducted. One looked at the environmental impact of the greenhouse study that I just talked about, and one looked at the environmental impact of a theoretical scaled up field study that was 1 acre or a little bit over 1,200 square meters in size. Steps covered within the LCA include soil amendment with fertilizer, seeding the species, cultivating the hyperaccumulator, harvesting the biomass, then washing, drying, and pyrolyzing the biomass to produce an end product, which was that rare earth-rich bio-ore. The emissions factor and global warming potential of the processes was also calculated. For the techno-economic analysis, that looked at the costs of the scaled up field study only, looking at the total costs of the process as well as the costs of materials and utilities. So results from the phytomining experiment indicated that P. arundinacea, or that common name reed canary grass plant, was the best hyperaccumulator, accumulating a little bit under 28,000 micrograms per gram of the mixed target rare earths. These were cerium, lanthanum, neodymium, praseodymium, and yttrium. And it exhibited the highest average bioaccumulation factor of 3.67. So looking at the statistical results for this one, let's look at success rate first, which is if a pot grew enough biomass to actually be characterized. The success rate was most positively impacted by soil treatment with fertilizer and non-treated water. The dry biomass and bio-ore yields were most significantly impacted by soil treatment with fertilizer again, and this time with water treatment with citric acid. For the most part, species didn't really impact biomass or bio-ore yield, except for in the case of B. juncea, which produced the most bio-ore of the entire experiment. And then moving on to bioaccumulation factor here, which is if a plant is a good rare earth hyperaccumulator or not. This was only only significantly impacted by species, with the, again, P. arundinacea performing the best out of all the species tested. The life cycle assessments identified that the highest contributors to the greenhouse gas emissions were the electricity demand of the greenhouse in the greenhouse study, and then the electricity demand of the pyrolysis reactor for the field study. Impact categories that were affected by this process include global warming, smog, and respiratory effects. The techno-economic analysis showed that the total cost of growing P. arundinacea for 6 weeks on a 1 acre field is a little bit over $6,000 US, with the cost breakdown shown in this pie chart here. 39% of that cost was due to cultivation, 22% due to biomass processing, 21% due to soil treatment with fertilizer, and 18% of that cost due to the pyrolysis step. Conclusions for this section: soil amendment and water treatment significantly impacted dry biomass and bio-ore yields, while plant species selection most significantly impacted bioaccumulation factor. Phalaris arundinacea achieved the highest average rare earth concentration of the entire experiment. And this pathway of phytomining, then drying and pyrolyzing the biomass, offers a promising low emission approach for rare earth extraction from Idaho source soil, specifically from the Diamond Creek area. The Chapter 3 objective was to examine the survivability, biomass production, and rare earth hyperaccumulation ability across various species and varieties. And this was done through a 2-step greenhouse phytomining experiment. And currently the manuscript is under revision at the journal Science of the Total Environment. So as you can see from this picture here, there were two phases within this experiment, the survey study and the variation study. Starting with the survey study, plants were grown for 10 weeks from germination in the Idaho-sourced rare earth-rich soil from Diamond Creek. Pots were arranged in a complete random block design with 45 unique species that were selected for their ability to grow well in an Idaho-based climate. There were 4 replications of each species to total 180 pots overall, which is what is shown in the pictures on the right. Mature biomass was harvested, washed, and dried, pyrolyzed at 350 degrees Celsius for 15 minutes. The resulting bio-ore was acid digested by the EPA 3050B method, and digestates were characterized via ICP-MS. For the second phase, which was the variation study, plants were grown for 6 weeks from germination in the Idaho-sourced Diamond Creek rare earth-rich soil. Pots were again arranged in a complete random block design, this time with 2 unique species. The Phalaris arundinacea, which was the top performing species of the Chapter 2 research, and then this Pseudorenaria spicata, which was the top performing species of the survey study that I just covered. 14 distinct varieties, 7 from each species, were randomly selected to be compared. This was to see if there was any difference between accessions in ability to hyperaccumulate and to produce biomass. There were 5 replications of each variety to total 70 pots overall. Biomass, again, was harvested, washed, and dried. This time it was ashed, so heated in the presence of oxygen at 350 degrees Celsius for 15 minutes. Resulting ash was acid digested using the EPA 3050B method and characterized via ICP-MS. So going back to the survey study for a moment, the Idaho native grass Pseudorenaria spicata, common name blue bunch wheatgrass, performed the best out of the 45 species tested. It had the highest survival rate at 100%, an average bio-ore mass yield of 4.1 grams, and a total mixed rare earth content of a little bit under 8,000 micrograms per gram of dried plant tissue. So looking at results for the variation study, as you can see from this table, the Phalaris arundinacea performed better than Pseudorenaria spicata in virtually all categories. It had a higher survival rate. It produced more biomass for both the root tissue and the shoots. And it accumulated more mixed rare earths in both the roots and the shoots. However, there was one variety of P. spicata that accumulated the highest average total shoot rare earth content of the entire experiment, accumulating a little bit over 36.5 thousand micrograms of mixed rare earths per gram of its dried shoot tissue. So looking at the statistical analysis, species was found to have a significant effect on root, shoot, and total dry biomass yield. There was also some effect of variety on biomass yield, but that effect was a lot stronger for the P. spicata species than for the P. arundinacea. Plant rare earth content, however, was most significantly impacted by tissue type, with the roots accumulating far more than the shoots did. There was also a significant effect found in terms of cerium accumulation, so species positively impacted cerium accumulation, and there was no other effect of species on rare earth accumulation ability, and there was no effect of variety at all. So these charts show that both species had really similar internal rare earth compositions, with cerium, lanthanum, neodymium, and yttrium being present in levels far higher than any of the other elements that were characterized. And interestingly enough, results showed that lanthanum was more abundant in the plants than it was in the soil, which was not a pattern seen for any of the other rare earths that were characterized. So to conclude for this section, all varieties of P. spicata and P. arundinacea performed similarly with regards to rare earth accumulation. There were significant effects of species on biomass yield and also some effects from variety, although those results were stronger for P. spicata than they were for P. arundinacea. Previous evidence describing P. arundinacea as a rare earth hyperaccumulator, such as my chapter 2 research that I just talked about, has been supported and strengthened, and the rare earth hyperaccumulation efficacy of P. spicata, which hadn't been explored up until this point, has now been studied and documented. So overall research contributions of the thesis. It evaluated the feasibility of phytomining in Idaho using various plant species, treatments, and of course that rare earth rich soil sourced from the Diamond Creek area. It compared the rare earth hyperaccumulation ability across different species and varieties. It assessed the environmental impacts and associated costs of Idaho-based phytomining through life cycle assessment and techno-economic analysis. It screened previously untested plant species to check for their rare earth hyperaccumulation ability. And it explored the applications of machine learning and AI within the field of phytomining. So future research opportunities include screening more species for rare earth accumulation ability to expand the current hyperaccumulator database, especially focusing on plants that are native to the Idaho area. Addressing challenges such as low survival rate, biomass production, and translocation efficiency, which is the ability of the plant to move those metals from the roots into the shoots. And addressing these challenges through methods like genetic engineering, selective breeding techniques, and the use of performance-enhancing treatments. Integrating machine learning and AI to improve and optimize all steps in the process, as well as use it for sustainability and economic modeling. And utilizing life cycle assessment and techno-economic analysis to provide more information on the environmental impacts and costs of large local operations. So now I just want to take a few minutes to highlight some of my accomplishments I've had since starting my master's program a few years ago. Again, I want to talk about my articles and manuscripts that I have out. So my chapter 2 manuscript was published in Sustainability back in June earlier this year. My chapter 1 manuscript is under revision at the journal Renewable and Sustainable Energy Reviews. And my chapter 3 is currently under revision at the journal Science of the Total Environment. Last year, I also had the opportunity to create my own undergraduate level course titled Phytomining, Emerging Biology, Technology, and Sustainability. And this course was actually offered this past summer, 2025, through University of Idaho as a 400 level 3-credit course. Other accomplishments. I won a pretty significant cash prize from the student poster competition at the Idaho Mining Association conference last year. Some of the more unique skills I've gained have been the procedural design and safe execution of acid digestion, and the operation and troubleshooting of a pyrolysis reactor and ICP-MS unit. So the picture on the top shows me working in the hood, digesting the bioores through the EPA 3050B method. And the bottom picture shows me taking those resulting digestates and running them through the ICP-MS unit. Funded projects that I've been a part of the past few years include this Idaho Global Entrepreneurial Mission funded project that tried to find novel ways to mine rare earths. And this is actually what funded all of the research that I've presented to you today. I also had the opportunity to work on a National Science Foundation funded project that uses education initiatives to make AI and machine learning more accessible to the public. Finally, I just want to finish with acknowledgements. First and foremost, I want to thank my committee members for being here, Dr. Merkley, Dr. Dolman, and Dr. Hong. I also want to thank my co-authors, contributors, and supporters, Tony, Ying, Ellie, Ethan, Sneha, Dave, Alice, and of course my family. Everyone on this slide helped me get to where I am right now, and I could not have done it with your help every step of the way. So thank you very much. Right, questions?
Thank you.